Welcome to all of you to this third lecture of nucleic acid chemistry. In this lecture, we will be discussing the structure and composition of the RNAs, different types of the RNAs, particularly focusing on the mRNA, tRNA, rRNA, and an introduction to small nuclear ribonuclear protein and microRNA. As we know from the previous lecture that RNA is another nucleic acid in addition to the DNA, and it is a polymer of nucleoside monophosphates. It contains the four different kinds of bases that are adenine, guanine, cytosine, and uracil here instead of thiamine. And this RNA is synthesized from the DNA directly with the help of an RNA polymerase. This process is called as transcription process. But unlike DNA, RNA is rarely composed of two strands. Mostly it will be existing as a single strand entity, as you can see in the accompanying picture. And usually when the double helixes will be formed in the RNA itself, it will be during the folding process in specifically rRNA and snRNA. In nature, 11 different kinds of RNAs have been found, but three of them are predominant because they help in the decoding of the genetic information in the DNA. And this sequence of bases in the DNA is then translated into a sequence of amino acids in proteins. The three types of RNA which are involved in translating this genetic information are the messenger RNA called as mRNA, transfer RNA, tRNA, and ribosomal RNA called as rRNA. There are other two important RNAs also. This is called as small nuclear ribonuclear RNA, which is used in the processing of the mRNA in cyclosome, and microRNA, also called as mirs or miRNAs, which are useful in regulating the expression of the genes as well. First important type of an RNA which helps in the decoding of a genetic information in DNA is the messenger RNA. It is synthesized directly from the DNA. It carries the genetic information from the DNA sequence to the ribosomes, where it tells the ribosomes to make a protein of specific amino acid sequence. Hence, we can say that mRNA serves as the mediator of converting the genetic information into the sequence of amino acids in proteins. The information which is present in the mRNA is actually got from the template DNA during the transcription process, and then it carries this information as a set of triplet nucleotides which are called as codons. Codons are the ones which specify specific amino acid during the translation process so as to drive the sequence of amino acids as according to the sequence present in the DNA. I have covered it in the molecular biology lecture, what are codons and how they specify specific amino acids. You can go to that lecture and see to it in depth. tRNA is the another molecule, RNA molecule, which helps in the decoding the information. It serves itself as the adapter molecule for the translation of genetic code. And in specific, the tRNA, as you can see in the picture, it has two important arms which are necessary during the translation process. One is called as the amino acid acceptor arm, another is called as the anticodon arm. This anticodon arm will crosstalk with the codon of the mRNA in decoding its information, and the amino acid arm is attached to a specific amino acid, which is actually the function of this anticodon. So during the translation process, the anticodon forms the complementary base pairs with the codons of the mRNA and helps in the decoding of the nucleic acid sequence in the sequence of amino acids of proteins specifically. In its structure, tRNA is a bit complex than the mRNA. While mRNA being a complete run of nucleotides only, it doesn't fold into a secondary structure. tRNA needs to fold into a secondary and tertiary structure for its function. In its composition, it is made up of around 74 to 95 different nucleotides, and about 20% of which are modified ones, which are modified after it is synthesized from the DNA. Around 50 different types of modified bases have been observed in several hundred tRNAs which are characterized so far. And out of these 50, these few are the ones which are most commonly found in almost all of the tRNA structures: pseudouridine, ribothymidine, dihydrouridine, inosine, specifically methylcytidine, methylguanosine. They specifically give the identity to the each kind of a tRNA, sort of as a face to the tRNAs in identifying the different tRNAs differently. So every tRNA will have a specific composition of a modified base and a specific site at which this modified bases are, so as to allow identification of a separate tRNAs separately. So all tRNAs will have a common secondary structure which is formed due to the complementary base pairing. The same rules which are followed by the adenine versus thymine and guanine versus cytosine. The first structure which they form is the secondary structure, which is also called as clover leaf structure of the tRNA. As you can see here in the picture, this is the clover leaf model of the tRNA, which is the secondary structure formed due to complementary base pairing. Discussing some of the important features of the clover leaf structure, we will see that this clover leaf structure of the tRNA, which is a secondary structure, has 5 distinct parts, 2 of which are the important in translating process. These 5 parts are the amino acid acceptor arm, then the anticodon arm, which are almost opposite to each other. Then the three things which give the distinct identity to the tRNAs is the D arm, variable arm, and the TYC arm. All of these arms are consist of 2 different things. One is the few base pair stem, and then there is the loop attached to this stem. So here, if you can see in the D arm, there is the stem of some base pairs and then the looping. So every arm will almost have it. So discussing the amino acid acceptor arm, which is one of the primary secondary structures of the clover leaf structure of tRNA, it possesses invariant residues of 74 to 76, 74, 75, and 76, which are added to it after the synthesis process. This is the post-transcriptional process by which it is formed in the 3' end of the tRNA for doing its function, especially in eukaryotes. It is the site where the amino acids are bound to the tRNA. That is, when the amino acid bind to the tRNA at this site, the tRNA is called as charged tRNA. And it's a whole different kind of a process which I have covered in the molecular biology. You can go and see those lectures in depth. The tRNA mature 5' end is also synthesized after the synthesis process. It's also a post-translation process, and it's formed by the RNA's P cleavage. And this arm also contains a 7 base pair stem, by which it is characterized in the length. Another important part of this cloverleaf structure is the anticodon arm, which is almost opposite to the amino acid acceptor arm. It's made up of 5 base pair stem and a 7 base pair residue loop. This 7 pair residue loop contains specific triplets, which are called as anticodons, as I already told you. These anticodons make a complementary base pair with the mRNA in reading it. So anticodon base pairs with the codon triplets in reading during the translation process. So this arm is also important. And anticodon arm is the one which gives distinct identity to the tRNA in charging it with a specific amino acid. So amino acid charging is based upon the type of an anticodon and the anticodon, what anticodon specifies. Then the three other structures are the D-arm, which consists of 3 to 4 base pair stem, and D-loop, which contains this modified base, which is dihydrouracil. Similarly, variable arm is called as variable because it can vary in length depending upon the type of a tRNA. It's between 3 to 21 residues and may form a stem up to 7 base pairs in length. Then there is third thing, which is the T-Psi-C arm, which is composed of 5 base pair stem and is ending in a loop which contains a specific modified bases GT-Psi-C. These three are also important in giving the 3D structure to the tRNA, which is the next level of folding for the tRNA in its functional form, especially the D-arm. D-arm is mostly involved in the during the folding of this tRNA into a 3D structure. Tertiary structure of the tRNA is the highest level of its folding, which is necessary for it to do its function of decoding the information during the translation process. As you can see in the panel, it looks like a letter L with two different ends. One end is the amino acid acceptor arm containing the amino acid, and another is the anticodon arm, which is necessary for reading the codons of the mRNA during translation in the ribosomes. This structure is actually stabilized by 9 different hydrogen bonds which are formed between the different structures of the cloverleaf model which you just went through. What are these 9 different bonds? These 9 different bonds usually involve the DHU loop and its arm and helps in the folding of the D arm over the TYC arm. Two important ones are the bonds between the DHU loop and the TYC loop, which are the modified G base and the Psi, and between G and C respectively. As you can see in the accompanying picture, I have given you these bonds which are present in the tertiary structure. And another one is the one with the TYC loop between T and A here. The another one is between the anticodon loop within it, between the Y modified base and a purine here. Then the rest of the 5 bonds are between the DH arm and the variable arm and within it. 3 are between DHU loop and the variable arm, and 2 are within the D arm itself. These are the 9 different bonds which help in making the folding of the tertiary structure of tRNA possible. And in its tertiary structure, tRNA is able to function only. So finally, we reach to the third RNA type, which is necessary for the decoding of an information and in synthesis of the proteins, is the ribosomal RNA itself. Ribosomal RNA, also called as rRNA, is the stable form of the RNA among all these three types. Why? Because they are the ones which make up the seeds of the protein translation. They make up the ribosome in itself, and it is here where the translation of the protein takes place. It's the stable and has got the highest half-life, and in comparison between the prokaryotes and eukaryotes, you will see the complexity and the availability of the more proteins in the eukaryotes than the prokaryotes. If you take the prokaryotic structure of the ribosome into consideration, you will see that the molecular weight is almost half than that of the eukaryotes, and it is also made up of 2 different types. One is called as the 50s, which is called as a large subunit, and the another subunit is the small subunit 30s. Together they make the 70s ribosome in the prokaryotes. And this large subunit in the prokaryotes is made up of 2 different types of rRNAs, that is 5s and 23s rRNA, and together with 36 proteins it makes up this 50s subunit, while as 30s subunits is made up of only one type of an rRNA and 21 proteins. When you compare these assemblies with the eukaryotes, you will find not only the increase in the molecular weight but also the increase in the availability of the RNA part. The eukaryotic large subunit, which is 60S, is containing three different types of rRNAs: 5S, 28S, and 5.8S rRNA, while as the small subunit is made up of 16S RNA, together with the 33 proteins, and the higher 60S is containing 49 proteins. These RNAs in the eukaryotes are synthesized by moderately complex DNA, which I have covered in the molecular biology section. You can refer to that also. So this panel gives you the overall visualization of the complexity as well as the content of the different subunits of the bacterial versus eukaryotic ribosomes. Another important type of an RNA is a small nuclear RNA, which makes up a cyclosome in the eukaryotic cells. In the eukaryotic cells, this is a part of cyclosome which is necessary for the processing of the immature RNA transcript into a mature RNA transcript. This cyclosome or snRNA helps in the removal of the non-coding sequences that are called as introns from the immature mRNAs, resulting in the formation of a mature RNA after splicing. We have covered this in the molecular biology as well, how the process of splicing takes place. MicroRNA, as the name suggests, are the smallest types of RNAs available in the nature. They are usually containing a sequence of about 22 nucleotides only. They are found in plants, animals, and viruses found for their RNA silencing or post-transcriptional regulation of expression of the gene in itself. It is one of the way by which the RNA controls the expression of an mRNA. So microRNA tells a mRNA whether it has to be expressed in proteins or not, or it should be arrested. As you can see in the accompanying picture, the small microRNAs usually get associated with the mRNA, which is to be transcribed and prevent the transcription process. So this is one of the function of the microRNA. It forms a complementary base sequences with the mRNA and hence prevent it from the translating and hence regulating the expression. This is one of the important functions of the microRNAs. This finishes the nucleic acid chemistry altogether. Do visit my Amazon page for the books which you might like and might find beneficial. And do subscribe to my channel for further videos which I will upload in due time.